conference this morning for the UCD Institute of Food and Health Research Bites. So my thanks this morning to Professor Sonia Negrao, and she's going to speak to us about her research in the area of heritage barley. Now, Sonia graduated in agronomic engineering, and she majored in crop breeding at the Instituto Superior de Agronomia from Lisbon in Portugal. She then went on to do her PhD in biology at ITQB, Nova, also in Portugal. And there she continued her work on breeding Portuguese heritage rice lines, and she did this in combination with the International Rice Research Institute of the Philippines. She then went on to do her postdoc, continuing her expertise in the area of rice genomics, and took a trip to far-flung parts, working in the King Abdullah University of Science and Technology in Saudi Arabia. And there she continued her work on the genetic dissection of the mechanisms of salinity tolerance in different species. We were delighted that Sonia joined the team in UCD as an assistant professor in 2018, and now she's the principal investigator of the crop stress interaction group. So over to you, Sonia, to share some of your work with us. Thank you very much.
Thanks, Dolores. Thanks for the nice introduction. So my research focused on one big question, which, as you can see, this is an old photo from the Rice Research Institute on submergence rice. And the question that my group poses is, why can some plants tolerate stress and others cannot? So in a nutshell, what are the genetic mechanisms that enable some of these lines or varieties to tolerate stress and cope with it? So when I came to Ireland, the first thing that I thought was about Irish pubs, Guinness, and whiskey, which I love. And for that, there's one big thing in common, and that's barley. So barley is essential for the malting industry. Barley is the fourth most important cereal crop in the world, and it's the first cereal crop in terms of importance in Ireland. It can be used for human food. So for instance, if you, the Irish stew has barley, it's also used for animal feed, and of course, very importantly, the production of alcohol. So for brewing and also for the malting industry. And we all know that climate change is impacting our food. So you can see from these images, drought, flooding, but also different diseases that are coming up in areas that before they didn't exist. So with this, we go to the Sustainable Development Goal 13, which is climate action. And as you can see here, from this photo, this was taken last week or 2 weeks ago in Glasgow at the COP26. So we clearly are in a climate emergency, and we clearly need to address it. And so we have to be ambitious to do so. So from the FAO, we know that the crop yield between 2005 and 2015, more than $9.5 billion were lost, particularly to flooding. So I'm focusing on flooding because, as you know, climate change is affecting rain patterns. So we have extreme weather events, we have floodings, but then we have droughts as well. So we have an unpredictable weather that can happen throughout the season. And as you can see here, you can clearly see from these images how the number of flood events have dramatically increased in the last few decades. So climate change is affecting barley production, and these are some photos of flooding fields here in Ireland. So this photo over here is from the Waterford distillery in 2019. There was a rain during the spring, after the sowing, and as you can see later on in the season, nothing grew here, and it really affected the production. So plants cannot run away from stress. They are they are there. So they have to cope with it with abiotics, different types of abiotic stress, and also different types of biotic stress. And to do so, because they cannot run away from stress or go inside and put a jacket or drink water when they are thirsty, they had to come up with very specific genetic mechanisms that enable them to be resilient and to cope with the stress. So when we are talking about waterlogging, which is what I'm focusing on, we are talking about when the water affects only the root area. So flooding is the term for everything. So waterlogging is only in the root area. So the aerial part of the plant, the shoot, is not affected. And when we are talking about submergence, we are talking about either the complete submergence of the plant that happens a lot in rice. It's a major problem in rice, but also a partial submergence when you have part of the stem or the shoot being affected. And what happens is that the soil spaces are replaced by water, meaning that you then get into hypoxic conditions or even anoxia. So there's no oxygen in the root area. And this means that there's an energy crisis because the plant, the roots are not performing respiration. So it goes into different processes and even fermentation to get the energy. So in a nutshell, this is what we observe is that under normal conditions, you have oxygen in the soil, you have the redox conditions are fine, you have a balanced uptake of the nutrients. However, under waterlogging conditions, you have way less oxygen or even not oxygen at all, and the nutrients uptake is affected. So for instance, some nutrients can become toxic, such as iron, but on the other hand, you have nitrogen and potassium being less uptake because of the change in the redox potential. So how does the plant respond? So there's a lower biomass, there's a reduced branching, there's yield, there's the leaf coloration is affected, so there's a chlorosis, the stomata closed, and the most classical example is the formation of erenchyma, which are basically shallow pipes caused by programmed cell death that then lead the oxygen from the shoot to the roots so that the roots have oxygen. So how can we assess this plant performance? How can we then look into the plants and try to understand what they are telling us? So the best way to do that is by phenotyping. So we know that the phenotype is the product between the genotype and the environment and the interaction between the two of them. And you can have the same genotype. So for instance, if you have exactly the same Arabidopsis plant, if you grow it in the short day conditions, you have a phenotype. If you grow it in a long day conditions, you can have a totally different phenotype. So the interaction between genotype and environment are very important. So how can we look into plant phenotyping, which is where I try to do my best. So you have the classical way, which all of us have done in our undergrads and even in our graduate studies. So there's a destructive analysis. We cut, we harvest, we look into yield. We have a sort of a snapshot, if you would like, of what's happening in that moment, okay? So it's usually a destructive analysis, the classical phenotyping. Now there's the high throughput phenotyping, and high throughput phenotyping, which is where I'm focusing on, involves cameras, involves imaging sensors, different types of cameras. And because we are taking photos throughout time, it's non-destructive. And you can follow exactly the same plant throughout time, meaning that that's extremely powerful when it comes to genetics, because the genetic signals will also change throughout time. So some genes can be responding to a specific time, and 10 days later could be another set of genes responding to the same stress. So this is extremely powerful in terms of genetic analysis and, for instance, association mapping. So when we are talking about classical phenotyping, you can see two of my students here. At Rosemount, you can see that they were harvesting the plants and they were separating the roots and washing the roots. Then they would measure the roots, weigh the shoot, fresh weight, fresh mass, etc. We can also use handheld devices such as, for instance, in this case, the spot, which is a proxy for chlorophyll content. And of course, you can go to the field and have fruit and yield related traits, as you can see it here. Myself in Saudi Arabia harvesting tomatoes, or a PhD student of mine in Lyons harvesting barley. And of course, you can also do, and many researchers do, a visual scoring. So that's phenotyping in a classical way. It's a destructive process. However, we can use high throughput phenotyping, and we can use different types of imaging sensors that can retrieve the plant phenotype throughout time. So what I'm talking about today, very briefly, it's about my project, which was funded by SFI. And the project aim is to elucidate barley stress adaptation in the context of climate change using disruptive technologies, in this case, imaging sensors. So it's an interdisciplinary research. We are doing high throughput phenotyping under field conditions, and we are using drones for that. And we are also doing it under controlled conditions at Rosemount with the equipment that I'm just going to mention, and also with other collaborators in France. We are using machine learning because this becomes a big data problem to analyze these images. And then we have genomics and genetics. So we genotype a collection of barley so that in the end we do association mapping and we can dissect the genetic basis of waterlogging tolerance. So genetic diversity is key. You can see this photo behind me, and you can also see this beautiful photo showing the genetic diversity. So we assembled a collection called the exhibit. I'm just going to mention in a minute. And so this is comprised mainly of heritage barley. So heritage barley is the barley that comprises both land races, meaning that they were selected throughout time by farmers, but also formally bred cultivars that had the hybridization process, breeding process, mutagenesis, whatever, and all of them have been extensively used in the past for production of barley. And we hypothesized that this barley is more resilient to stress just because it has a better resource use efficiency, as it was bred most of these lines before the Green Revolution. So here is just a photo of the spikes of our barley, of the European Heritage Barley collection, to show a little bit of the genetic diversity that we have in the collection. And so the exhibit collection comprises basically Northern European lines, 2-row spring barley, and it's comprised of 365 accessions. Most of them are cultivars released before 1990s, and we also have some land races, and you can see that's more throughout in Northern Europe. Why? Because it's in Northern Europe that we would have higher probability of flooding events and resilience that was built throughout time. If I would be looking at drought, I would probably be looking into South European countries. So we genotyped the entire collection, these 365 lines, with 50K iSelect SNP array. We performed structure, and it seems like we have 4 groups being suggested by the softwares. So what we tried here was to do we see a sort of a pattern? So on the left side, we try to see if there was a geographical pattern, and you can see that yes, you have a sort of like northern lines tend to group in a different group over here together with more western lines, but then you have a little bit all over Europe. On the other hand, if we group by year of release, you can see that we have a sort of a trend, that we have two major clusters here, before 1990s and after 1990s. The cultivar release, you can have a different group. So it seems like we probably have a population structure depending on the cultivar year released, and not by geography. So what did we do next? So we assembled the collection, we genotyped the collection, we looked at the population structure. So now we need to phenotype. We looked at the genotype. We now need to look at the phenotype. So we did that in controlled conditions. So this is a photo in Rosemount, and also we phenotype in field conditions at Lyons Farm. So we have this imaging platform coming from Photosystem Instruments at UCD that comprises these three modules. And just going to explain a little bit first. What we did was that we need to optimize the protocol. So we didn't know what was the best protocol, so that was the first step. We picked up two Barley lines, one heritage, one elite line. So one Irish line that was probably what your grandparents or parents would drink, Guinness back in the 50s or 60s, and then an elite line, which chances are if you're drinking a pint of Guinness, it's coming from this line. So we did control versus waterlogging, and we tested different stress periods, 5 days, 10 days, and we tested different recovery with or without recovery. So this is just a short video to show. So this is how we tested the protocol. We did a split plot design where you have the control line next to the waterlog line. And you can clearly see that we have the image here. So this is how it goes. You have a QR code that's red, and the conveyor belt moves the tray inside, and then the image process starts. And of course, we then measured the end period so that we could correlate what we would see with the imaging sensors with real biomass indicating in a process that's named ground truthing. So this is an image of what would happen if we would just look at the photo on the top. So this is just an image. And what we have on the system is that we have three different types of sensors. So the first sensor is a chlorophyll fluorescence sensor that enable us to look into photosynthetic traits such as quantum yield or Fe over Fm. Then the next sensor, so each here you have a door that opens and moves on through the conveyor belt through this system. We have an RGB image, which allow us to look into growth and morphometry because we are following again the same plant throughout time, so we can actually see it grow. And then we have hyperspectral, which enable us to look into stress indices and some pigments such as carotenoids. And what we looked at is that this preliminary data, and this is just an example, and again, this is preliminary data, is that we look into Irish accessions. So this is the PhD from Patrick Langan. And you can clearly see that when we are looking into biomass over here, you have the protocol. So in orange is the stress period, and in green is the recovery period. And you can see how the plants were growing in control conditions in the full line and in waterlogging conditions with the dashed line. So you can clearly see that there's a very distinct growth arrest under waterlogging. And you can see it that it starts probably in some plants after 4 days or even early. It really depends on the genotype. For instance, this genotype, you can see it seems like it only later starts to have this growth problem. And with the hyperspectral camera, we can see clearly that after the first day, you have a major drop on the photochemical reflectance index, which is a very strong proxy for stress response. So all the lines, and then they start recovering throughout time. So this is only preliminary data. And why do I say it's preliminary data? Because we are having some issues. So one of the major issues is called image segmentation. So I showed you the photo of the plant. Okay, so this is the raw image of what the system takes the photo, but then you have to segment the image, meaning that you have to extract the green pixels from all the rest of the background and pieces of equipment, tray, etc., which is basically doing this. Okay, so this is the segmentation. Now the segmentation is working quite well for the RGB camera. However, the hyperspectral camera, we are having some problems. So this is a hyperspectral cube. This would be the image that would be, we are putting hyperspectral cube because you cannot, it's not a 2D image, it's a 3D image where each of these layers is a wavelength, and the hyperspectral image is analyzing more than 140 wavelengths each image. So each of these layers is one different wavelength. And so what the PSI system default system does is just segmentation. This is the segmentation that it offers. And you can immediately see the problems. The overlay, the overlap of the object with the segmentation is not perfect. So we are, and it's also capturing background noise here from the equipment, and it's counting as green pixels, which they are not. Okay, so the accuracy is actually quite low. So one of the PhD students in my group, Jason Walsh, so he's a computer scientist, and he has been working on the segmentation using machine learning. And when he started working on that in deep learning, so this is a deep learning method, he got into the accuracy of 0.98, and you can clearly see that now the image is overlapping quite well, and you're not capturing extra pixels from the equipment. So in the field, we did a typical process. This is what we had from last year's data. So you can see a very nice histogram. So we have a very good distribution of the data in these 365 lines, but that was too hard for us. So we lowered down to a core collection that's here in yellow, which is 230 lines. And this is what we have been now using moving forward on the project. So you can clearly see how the behavior in terms of different yield parameters. And we also optimized the stress last year to try to see what was the best way of stressing the plants and waterlogging them. And so we saw that the soft spot was around 2 to 5 days that we would get nice results in terms of yield. We would have an effect of waterlogging in the plant performance, but without putting too much time and water resources, which is absolutely crazy. So this is just a short video of this year field trial. So over here, you can see the control plot. So all of these, each one of these is a plot, individual plot, and then you can see it here, the waterlogging plots. And this is just a very short video of the aerial view of Lions Farm of what we had. And you can also see here the containers for the irrigation and the tubes for the irrigation. So how was the logistics of the irrigation this year? So we, first going to show you the images of the control throughout time. You can see the plants growing and the plots growing throughout the season. And then the irrigation logistics. So we used 2 tanks. Of each tank had 26,000 liters. We had 1,700 meters of pipes. So the tanks had to come by lorry. So when you see all the pipes here, we had an electric generator, 2 water pumps to actually manage to soak the soil in this area. And believe me, we reduced as much as possible we could for the requirements of water to get the approval and all of that. So after 4 days of irrigation, we ended up doing 4 days of irrigation, 12 hours per day, with a total of almost, it's a little bit less than that, 250 cubic meters of water. So this is what you see. You can clearly see the water going up and just reaching the soil saturation and then dropping once we switched off the taps and going down. And it was actually a very wet season as well. So then we processed the drone data. You can see here the control plots, the water logging plots over here. So the NDVI. So we are now on the process of getting all this data nice and neat. And you can see the water logging trials as well progressing throughout time. How do they look like? And what we see, this is again preliminary data, it's still not spatially corrected. You can clearly see a massive reduction in several traits from control to waterlogged. So we can clearly see that the 4 days of waterlogging were enough to cause a shift in all the data, and also delaying the flowering time and the heading date. And so what are we doing now? We are processing basically all the data. So all the drone data, we are spatially correcting the field data from this year. And then we are performing agronomic analysis, agronomic data, so that we can look into an association analysis and exhibit statistical power that can be used in the field. And then of course next year we will do it all over again. Irrigation, flying drones, getting lorries with massive tanks of water to get everything done. And so there's lots of people contributing to this project. My team over here, everyone from the research assistants that have been doing a tremendous work getting all the data, but also the PhD students, the postdocs, and also Lions Farm, Eddie Jordan, Eugene Brennan. Without them, this would be impossible to do so. And then Eleni from computer science, and then Kevin McDonald's team as well that have been assisting us as well. The James Hutton, because all the assembly of the germplasm was myself and Robbie's team, and the University of Picardie, where we have our French collaborators that will phenotype the whole collection. And yeah, all those beautiful images were acquired by Drone Services Ireland. And thank you all, and I welcome your questions. I'm going to stop sharing here.